Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different aspects of the immunology in this particular module. So far what we have discussed about the basics of immunology and how the immune system is protecting the host from the different types of external antigens or the internal antigens, right? So the purpose of the immune system is to resist against any kind of changes into the host body, right, which is being induced by the internal factor or the external factors. Now, in this context, we have discussed about the basics of the immunology in the previous lecture. Now, in today's lecture, we are going to discuss about the antibodies and how the antibodies are going to be used for different types of applications of the immunology. So antibody is a Y-shaped structure, right? And it is been produced from the B cell, right? So remember that when we were in the previous lecture, we discussed about the humoral response, which is being mediated by the antibody. So first, let's discuss about the antibody structure and the classification. So antibody on stimulus with the antigen, the B cells secrete the antibodies with the antigen site identical to that onto the B cell antigen receptors, right? The identity between the binding site of the secreted antibody and the membrane bound B cell receptor was first demonstrated by making a reagent that bound to the antibody secreted by a particular clone of the B cell, and those reagents also found to be a receptor on the cell that secrete the antibody. So secreted antibodies and the membrane bound receptors belonging to the immunoglobulin family of the proteins, right? The large family of protein and this is a large family of protein which include both the B cell and the T cell receptors, addition molecules from tyrosine kinases and the other immune receptors. And it is being characterized by presence of one or more immunoglobulin domains. The immunoglobulin domain is generated when a polypeptide chain folds into an organized series of anti-parallel beta-sheeted pleats, right? And with each domain, the beta strands are arranged into a pair of beta sheets to form a tertiary compact domain. The number of strands per sheet varies among the individual proteins, right? The antibody molecule, the most immunoglobulin domain, contains approximately 110 amino acids. Each beta sheet contains 3 to 5 strands. The pair of beta sheets within each domain are stabilized with respect to one another by interchain disulfide bonds. Neighboring domains are connected to one another by a stretch of relatively unstructured polypeptide chains. So within the beta strand, the hydrophobic and hydrophilic amino acids alternate and their side chains are oriented perpendicular to the plane of sheets. The hydrophobic amino acids on the other sheets are oriented toward the positive sheets, and the two sheets with each domain are therefore stabilized by the hydrophobic interactions between the two sheets as well as by the covalent disulfide linkages. So remember, in the antibody structures, you are going to find a lot of covalent interactions mediated by the disulfide bridges, right? So you are actually going to have the disulfide bridges between the heavy chain and between the light chain as well, right? End of each of the beta sheets, more loosely folded polypeptide region, one beta strand to the next, and these loose folded region can accommodate a variety of amino acid side chain length and structure without causing any distortion to the overall structure of the molecules. And this is a very, very important feature, that it is actually accommodating the any kind of modifications, any kind of alterations into the sequence, right? Hence, in the antibody molecule, the immunoglobulin fold is superbly adapted to provide a single scaffold onto which the multiple different binding sites can be built, because the function of the antibody is to bind the antigens, and for that it has to change its structure, its charge distribution, and so on, right? These properties explain why the immunoglobulin domain has been used in many proteins with recognition or the addition functions. The essential domain characterized provide a molecular backbone, while the loosely folded region can be adapted to bind specifically to many adhesive or the antigenic structures. The immunoglobulin domain structure is used by the many protein besides the B cell receptors. The T cell receptor is also made up of the repeating unit of the immunoglobulin domains. The other protein that use the immunoglobulin domains are like Fc receptors, the T cell receptors, CD2, CD4, CD8 and CD28. Each of these proteins is classified as a member of immunoglobulin superfamily, and all antibodies share a common structure of the 4 polypeptide chains consisting of the 2 identical light chain and the 2 identical heavy chain, right? Each light chain is bound to its partner heavy chain by a disulfide bond between the corresponding cysteine residues, as well as the non-covalent interaction between the VH and the VL domain and the CH1 and Cl domain. So these bonds enable the formation of a closely associated heterodimer which actually contains the heavy chain and the light chain. Multiple disulfide bridges link the two heavy chains together about halfway down their length, and the C-terminal part of the two heavy chain also participate into the non-covalent interaction between the corresponding domain. The antibody molecule forms a Y-shaped structure with the two identical anti-binding sites at the tip of the Y. Each antigen binding region is made up of the antibody amino acids derived from both the heavy chain and the light chain and the amino terminal domain. The heavy and the light chain both contribute to domain to each arm of the Y chain, with the non-antigenic binding domain of the each serving to the extent to the antigen binding arms. The base of the Y consists of the C-terminal domain of the antibody heavy chain. The overall structure of the antibody molecule consists of three relatively compact regions joined by a more flexible hinge region. The hinge region is particularly susceptible to proteolytic cleavage by the enzyme which is called as papain. The papain resolve the antibody molecule into two identical fragments that retain the antigen binding specificity of the original antibody and the remaining region of the molecule, which consists of a non-antigen binding portions. The later region, which is identical to all antibody, is actually being called as the Fc region. So Fc region is not variable between the different antibody molecules, and that's why it is also been called as the constant region. Each FAB region and the Fc region mediate its own particular function during an antibody response to an antigen. The FAB region binds to the antigen, and the Fc region of the antigen coupled bind to the receptor on the phagocytic or the cytolytic cells or to the immune effector cells. In this way, the antibodies serve as a physiological bridge between an antigen present onto the pathogen and the cells or the molecule which will be ultimately going to destroy it, right? So a family of Fc receptor exists, with each Fc receptor is expressed on a different types of cells and bind to a different class of antibodies. So this is the structure of the antibody, where you have, this is the this region is the antigen binding region. So you have one antigen binding region on this side, and you have another antigen binding region on this side, and this is the light chain, right? So this is the light chain, this is the heavy chain, and you have the two light chain and the two heavy chain, which are actually been connected to each other by disulfide linkages at this junction, and this is being called as hinge region. On the other side, this is the region which is being constant between the antibody molecules, and this is being called as the Fc region. So this is the constant region, this is the variable region, and this is the region which binds the antigen. The heavy and the light chain in an immunoglobulin molecule contains the amino terminal variable region that consists of 100 to 100 amino acid and differ from one antibody to the another one. And the constant region exhibit the limited variation that defines the two light chains and the heavy chain subtype. The heavy chain, which is the gamma, delta and alpha, also contain a proline rich hinge region. The amino terminal portion corresponds to the V region, binds to the antigen. Effector functions are mediated by the carboxy terminal domain. The mu and the epsilon heavy chain, which lacks a hinge region, contains an additional domain into the middle of the molecule, which is called as CHO, defined as a carbohydrate linked to the heavy chain, right? So this is a typical structure of the antibody, right? And depending upon the different types of antibodies, it could be IgG, IgA, IgD, IgE and all that. You are actually going to have the composition of the different types of heavy and light chains. And now discuss about how the antibodies are being, different types of antibodies are being produced and how they are actually maturing and giving you the final antibody. Now, antibody diversity, right? So you basically have the different types of antigens. You have the antigens which has been derived from the bacterial sources. You have the antigen which has been derived from the viral sources. You have the antigen from the fungus, cancer. You have so many antigens, right? And accordingly, you should have different types of antibodies. So how we are actually producing the different types of antibodies? So multiple gene segments exist at the heavy chain, which is being called as V, D, and J. And you also have the multiple gene segment at the light region, which is called as V and J loci, and these can be combined with another to provide the extensive combination diversity. The p-nucleotide addition results when the DNA hairpin of the coding region is cleaved symmetrical, filling in the single-stranded DNA piece, resulting into this asymmetrical cleavage generate a short palindromic sequence. So exonuclease trimming sometimes occur at the VDJ and the VJ junctions, causing the loss of the nucleotide. Non-template N-nucleotide addition in a heavy chain results from the terminal deactivity, and that mechanism actually contains the 2,3,4 rise to give diversity at the junction between the gene segment, which contributes to the CDRs. So combinatorial diversity, the same heavy chain can combine with the different light chains and vice versa. Thus, there are 5 mechanisms are responsible for the creation of the diverse receptor of the B cell receptors and the antibody that is available to the organism before any contact with pathogen or the antigen occurs. Following antigen stimulus, the B cells are able to yet another mechanism unique to the immune system to further diversify and refine the antigen specific receptors and antibody and the somatic hypermutations. B cells are exposed to the successive cycles of the mutation at the BCR loci, followed by the antigen mediated selection in a specific region, selected a specialized region of the lymph node and to the spleen, and the end result of this process is the average affinity of the antigen specific BCRs and the antibodies formed at the end of an immune response is considered higher than its instigation, and this process is referred to the affinity maturation, which means what you are doing is you are actually generating a pool of antibodies, you are generating a pool of the B cell receptors, and then they are actually going to be selected by the interaction with the antigen, and based on this selection you are actually going to select the high affinity antibodies, and as a result that antibody containing clone is going to be proliferate and it will give you more antibodies. Then talking about the B cell development, the maturation, right? So in the bone marrow, the B cell developed by rearranging immunoglobulin genes like VDJ. So immature B cell expresses the IgM on their surface, right? So IgM is a kind of the antibodies where you have the, and so any B cell that binds self antigens are eliminated. So that is being called as the clonal deletions. Remember that you don't want our immune system to work against the antigen what is present in our body. So that is very important, because if you don't destroy those cells, they will actually going to act against our own cell, and that's how they are actually going to be responsible for the different types of autoimmune diseases. So surviving the immature B cell further co-express IgM and IgD and circulate through blood, lymph and secondary lymphoid organs like the lymph nodes and spleen. And when these molecules are going to encounter the antigen, right, then the antigen is actually going to start the selection process, right? So mature naive B cells encounter the antigen in the lymph nodes or to the spleen. Binding of the antigen to the BCR provides the first activation signal. So once the antibody is actually going to encounter the antigen, right? So it is actually going to give a stimulation signal, because you have the Fc region, and this Fc region is going to bind to the cell which is responsible for eliciting the immune response. And as a result of this cell is actually going to get the stimulus signal, and it is actually going to produce more amount of antibodies. It is also going to produce the other cytokines. So once this activation signal is being sent, then the antigen, a second signal comes from the CD4 positive T cell through the CD40 CD40L interactions and different types of cytokines like the IL-2 and IL-4. And then in the T cell independent antigens like the polysaccharides, activation can occur directly by the strong B cell receptors, cross-linking, or the innate immune receptors. Then we have the clonal expansion. So activated B cell proliferates, producing a population of the identical B cell specific to the antigens. Some differentiate early into the short-lived plasma cell into the primary foci, secreting the IgM, and the other migrates into the lymphoid and form the germinal centers. Then with what will be the process into the germinal cell centers? So in the germinal centers, the two key processes, the fine tuning and the antibody response. So in the somatic hypermutations, the activated induced cytokine enzyme introduces the point mutation into the variable region of IgG, generating the PCR variants, and all these B cells are actually going to be selected against the antigen, and those who are actually going to engage the antigen, they are actually going to be survived, and then they are actually going to start producing the IgG or antibodies. Uh, then you are actually going to have the class switching recombinations. So activation induced cytidine deaminase also derive the switch from IgM, right? Remember, this is the antibody what is going to be produced in the beginning to the other antibody class like the IgG, IgA, and IgE, giving the antibody the new effector functions, right? Because IgM has its own function, and IgG, IgA, and IgE are actually going to have its own other functions. Now, at this stage, there will be a memory formation and a long-term immunity. So the B cell becomes long-term plasma cells, sitting in the bone marrow and continuously secreting the antibodies. Other becomes some of these B cells are going to be transformed into the memory cells, so that they will actually going to keep a memory for this particular antigen. So as a result of this, when the same antigen will come to the next time, it is not going to go through with the extensive process of antigen processing, presentation, and so on. This antigen, as soon as this antigen comes, these memory cells are actually going to recognize this antigen, and then directly they are actually going to start secreting the antibodies, so that you can be able to have the early response, and that's how you can have the better protection. So once the memory cells are being produced, which respond rapidly and robustly upon future exposure to the same antigen. So antibody production starts with the naive B cells that upon antigen encounter an appropriate signal undergoes the clonal expansions, somatic mutations, and the class switching, ultimately generating the high affinity plasma cells and the memory cells. This refined process ensures a strong and adaptable immune defense. So this is all about the theoretical aspects, how the antibodies are being selected from the antigen, how the clonal selection happens, how the clonal proliferation happens, and so on. And as a result of that, you are actually going to produce the plasma cells, and you are also going to produce the memory B cells. But what happens when you are actually trying to develop a particular antibody, right? Suppose you want to develop a particular antibody against a particular antigen, then you are going to use either the different types of animals. You are actually going to use mice, rabbits, sheep, and goat. Even sometimes you use the horse also, right? So you might have heard about that the people are developing the different types of antibodies and the anti-sera against the the snake poison, right? So into the horse. And then you are actually going to utilize these antibodies for the different types of biotechnology applications. So to get you familiar with this whole process, how you are going to be generate the antibodies in a particular animal, we have prepared a very small demo clip where we are actually going to show you the salient features, how you can be able to prepare the antigens, how you are going to give the injections to the rabbit, and how you are going to collect the serum, how you are going to determine the presence of antibodies, and so on, right? So after this demo, probably you will be able to understand the whole process of the antigen production into the animals, and the similar kind of what we have discussed is what will happen inside the body. What we are going to discuss in this demo is about what are the things you are going to do so that the antibodies are going to be produced.
I am Amog Anand. I work in CSIR, CDR, I don't know. And in today's demo, we'll be discussing different steps involved in generation of antibodies in rabbits. So for the first step, we require several things like trans complete adjuvant. Here it is from Sigma. We need a microemulsifying needle which has two openings connected with a fine needle. We need antigen which is purified and filtered, so there are no contaminations. It is a sterile solution of antigen. Then we take out some of the Franz complete adjuvant in Nippendorf and then mix them together. Since this adjuvant is oil based, it doesn't mix easily with the watery system like the antigen is in previous. So therefore we mix them rigorously and forcefully. For that purpose, we take these two, we mix this emulsion and we mix this PBS containing antigen and the adjuvant, oil-based adjuvant. After mixing them, we take out in a needle. Using a needle, we take out in a syringe like this, and then we fix the microemulsifying needles into it. Attach another syringe into it like this. So once you have filled your antigen and adjuvant in this needle, you push it here and then you keep pushing from one side, keep pulling from another side, keep pushing from one side and keep pulling from another side. So this process forcefully pushes your material, I mean the oil and the antigen, through this fine needle, and with that, in that process, the emulsion is formed. Emulsion can be called as water in oil or oil in water because both are in the same concentration, same volumes, so you can call them anyway. By this method, the emulsion is formed. So for ready reference, we have already prepared the emulsion. This emulsion looks like white. Initially, it was two-phase, and then slowly it has turned into single phase. Now you can push this emulsion from one needle to another side and from another this syringe to another syringe. So this process creates very good emulsion which doesn't separate out later on when you are ready to inject. So how do you check them? So for checking purposes, a drop of emulsion on a water surface like this. If emulsion is not formed perfectly, this will spread out. Otherwise, it will not spread. So this is a check that your emulsion is formed correctly. So once you find that this drop is not spreading, your emulsion is actually ready for injection. So this was the process by which we prepare the emulsion for injection. So this is the first step of preparing the emulsion. So now let us understand why we prepare the emulsion. We have checked that emulsion is formed. Now the purpose of making the emulsion, because you have antigen and through antigen you can raise antibodies, but after emulsifying them, you actually make the antigen releases slowly. So it is a sustained release kind of preparation, so that the antigen is exposed to system in a systematic manner, so that more and more memory cells can is generated, and that's the sole purpose of having emulsion. Otherwise, if you inject antigen as such in PBS or in other watery system, it will spread out in the body and it will be cleared out by the immune system readily, and no memory cells will be generated. So this is the main purpose of preparing the emulsion. So now we have prepared the emulsion. We have come to animal house. This is our rabbit, which will be immunized, and before immunization, we have to take pre-immune bleed so that we can compare later the serum and the anti-serum. So we'll now start how we immunize it. So now we are preparing to immunize. The first and important thing is in all these animal processes is we have to avoid the pain to the animal. So for that purpose we have to strain the animal because we have to inject. So we strain the animal in a way that it has less and less pain and the movement is also less. So we'll inject this into the emulsion into the thigh. We have to catch hold of both the legs. Then we have to sterilize the area using alcohol. We have to look at the thigh muscles. They should be cleanly visible. Skin should be cleanly visible. There are two kinds of injection that we give. One is intradermal and another one is subcutaneous. So today we'll be doing subcutaneous injection. This is our emulsion that we have prepared by microemulsifying needle. We have seen right here. This is the area where we would like to inject. We have to take out all the airs from the syringe and the needle. We have taken out, clean the area again, and then we apply some antiseptic powder. Here it is betadine powder, so that the infection cannot develop later on. We sprinkle some of it at the area of injection, and then slowly release. Leave the animal relaxed and it is immunized. To strain the animal in a towel or this kind of cloth. So the advantage of having this cloth to strain is the animal has its claw outside of this cloth, and then it cannot move. So we have to restrict the movement. Pain is similar in the strainer and in the, on this cloth. So we use this bleach. Now we will strain the animal on this cloth. We keep the animal relaxed on this cloth and we will strain it. Make sure that the ear are outside and the animal is trained properly so as to reduce its movement, and now it is ready to bleed. We bleed the animal from this mid-ear vein and we rub it so that it gets loosened up and the circulation is faster. The vein will also expand and more flow will be there. So this is the method which is normally we apply. When the vein is properly visible, this is the mid-ear vein from which we bleed. We will slightly sterilize it using alcohol and using a 20 gauge needle which is wide enough to give sufficient bleed. We will prick the vein and collect the blood. to stop it, you just with less and less pain, you can collect the bleed like this. Now we have to make sure that no further bleeding occurs. Then we will wipe out whatever bleed is outside using sterile water. We wipe out all the blood here and there. If any spillage, we will wipe out all over using water so that the vein becomes cool and gets shrunk. You just check, still it is bleeding, so keep it pushed. Until the bleeding stops. Now I think the blood has stopped coming out, and then now we apply some antibiotic. Here in this case it is betadine powder, so that there is no further infections or inflammation in the rabbit, and this also ensures that this is your pinna, and if there is any inflammation, it will have some pain, so it will avoid that kind of pain also. So we have isolated approximately 10 to 12 ml of blood. This will give us approximately half of the volume of the blood serum. This will be coagulated, blood will be coagulated at 37 centigrade for 1 hour, and then it will be kept at 40 centigrade for overnight, so that the clot is shrunken properly and the serum is maximally taken out. And then we isolate the serum, add some preservative like sodium azide, and keep it at -20 or -80 as per the requirement. And then we also taste simultaneously the title of it and the specificity of it using ELISA test and the resting blood test respectively. Now you can see this whole of the animal, this animal is very relaxed. It has, I think, it has very less pain or a negligible pain. And this is actually very important for handling animal, that in whatever procedure you go through with animal should be ensured not to have pain. Maybe you can, if the procedure is painful, you understand it is a painful procedure, you anesthetize it. Since this procedure is not painful, you have not anesthetic. So that this is very important step to ensure that the animal has a pain. It should be relaxed. So in whole of the process, I think you have got to know all the steps of embryo development in rabbit, and we have prepared medicine. We injected the medicine. We isolated the blood after giving sufficient booster doses, and whole of the process is in the way. I think you have understood most of these processes, and you like it.
Now, once you have produced the antibodies, these antibodies can be used for doing the different types of antibody antigen interactions studies, and based on the antibody and antigen interactions, you can be able to develop the different types of techniques which can be used for different types of immunological assays, so that you can use them for diagnostic purpose. You can use that for detect identifying a particular pathogens, or you can use that for the identifying a particular disease and so on. So how the antibody and antigen interacts, right? So there are many factors which are actually going to govern how the antibody and antigen are going to interact, right? So antibodies or the immunoglobulins are the Y-shaped molecules which are being produced by the B cell. Remember, not the B cell but the plasma cell, right? Each antibody has a unique region which is being called as paratope that specifically bind to a particular part of an antigen which is being called as the epitope. So it is actually the interaction of the epitope and the paratope. The binding is highly specific and occurs through the non-covalent interactions, including the hydrogen bonding, electrostatic forces, van der Waals forces, and the hydrophobic interaction. The strength of the antigen and antibody interactions are determined by the affinity, avidity, cross-reactivity, and the specificity. So, in the affinity, the combined strength of the non-covalent interaction between a single antigen binding site of an antibody and a single epitope is the affinity of the antibody for that particular epitope. Then within the avidity, the strength of the multiple interaction between the multivalent antigen and the multivalent antibody is called as the avidity. High avidity antibodies are actually going to have the low affinity. Then you also have the cross reactivity. So an antibody produced for a epitope of an antigen also cross react with the epitope on an unrelated antigen, and this is called as the cross reactivity. So cross reactivity can occur because the two antigens share the chemically related epitopes, or the affinity of the antibody for original epitope is more than the cross reacting epitopes. Then you also have the specificity, the ability of an antibody to distinguish between the two different types of antigens. Now, based on the antigen-antibody interactions, people have developed the different types of immunological reactions, right? So, the first reaction is the precipitation reactions, where you can actually be able to use the antigen to precipitate the antigens, right? So, the interaction between the soluble antigen and the antibody is called as the precipitation reaction. The antibodies involved into the aggregation of soluble antigen are called as precipitants. The antibody, when the antibody and the soluble antigens are mixed in solution, the bi or the multivalent nature of the hemoglobin allow for a single antibody molecule to bind to more than one antigen. If the antigen is polyvalent, like for example more than one antibody binds per antigen molecules, then it will be binding the multiple different antibodies. Eventually, the resulting the cross-link complex becomes so large that it falls out of the solution in the form of a precipitate. Now, examples of the precipitation reactions, you can have the precipitation reactions into the solutions. So, a soluble immunoprecipitation can be used to purify the antigenic molecules from a heterogeneous mixture of the soluble molecule, or to remove the particular antigen from the solutions. Immunoprecipitation occurs only when the antibody antigen are essentially equivalents, and the antigen or antibody axis monovalent binding is favored and does not result into the formation of a precipitate. Then you can also have the precipitate reactions into the solutions. So immune precipitate can now can form not only in solution but also into agar mix. So when the antigen and antibody diffuse from one another to a visible line of precipitation will form. As in a precipitation reaction in solution, visible precipitate occur when the concentration of the antibody antigen are equivalent to one another. So immunodiffusion in the gel is a rapid, easy and easy to perform and surprisingly accurate. So in the the Outerloni method, the most frequent employed variation of gel immunoprecipitations, both antigen and antibody diffuse readily from the well each other, and thereby establishing the concentration gradient. And at the relative antibody interactions at which the lattice formation maximum turns to be equivalence, a visible line of precipitation or the precipitant line forms into the gel, right? So basically what you have is, you suppose you have two wells, right? So if in one of the well you put the antigen, another one antibody, so they will both are actually going to diffuse into the agar, right? And they both will interact, and as a result they are actually going to precipitate on the center, right? So they can actually be able to give you the line of precipitant, right? And this happens because at this point the antibody and antigen reacts in an equivalent amount, and as a result of that they are actually going to get precipitated, and they will come out of the solution and will give you a white color precipitate. More sophisticated analysis can give the information regarding the extent of cross reactivity and of the antibody preparation with the related antigens. Then we also have the agglutination reactions, right? So agglutination reactions are always been used for blood group typing, right? So you can actually be able to use the agglutination reaction for the blood group typing. So the cross linking that occur between the di or multivalent antibodies and the multivalent bacterial or other cellular agent result in a visible clumping of the complex formed between the cell bearing the antigen and the antibody molecule. This clumping reaction is called as the agglutination, and the antibody that produce such reaction are called as agglutinin, right? Agglutination reactions are identical in precipitation to the precipitation reaction. The only difference is that the cross-linked precipitate is visible to the naked eye because of the large size of the antigen. One of the simple example is that you are actually going to detect the antigens onto the surface of the RBCs in a assay which is called as hemagglutination. A bacterial infection often elicits the product of the bacterial antibodies specific for the surface antigen on the bacterial cell. Such antibody can be detected by the bacterial agglutination reactions. The principle of bacterial agglutination is identical to the hemagglutination, but in the case the visible pellet is made up of a bacteria cross-linked by the uh, antibacterial antibodies. So apart from these reactions, you can also use the antibody-antigen interactions to develop much more sophisticated technique, which is called as the enzyme-linked immunosorbent assay or the ELISA. So ELISA is an array for quantification of either antibody or the antigen using the enzyme-linked antibody and substrate that form the colored reaction products. The conjugated enzymes are selected based on their ability to catalyze the conversion of a substrate into a colored fluorescent or the chemiluminescent product. The number of variation of the basic ELISA assay have been developed. Each type ELISA can be used quantitatively to detect the presence of antibody or the antigen. Alternatively, a standard curve based on the known concentration of antibody or antigen can be prepared and be used to determine the concentration of a sample. So you can have the three different types of ELISA or four different types of ELISA. You can have the direct ELISA, you can have indirect ELISA, you can have a sandwich ELISA, or you can have the competitive ELISA. So let's talk about the indirect and the direct ELISA. So antibody can be detected or its concentrations determined with an indirect ELISA, right? So serum or the other sample containing the primary antibody is added to a antigen coated microtetraplate plate and allowed to react with the antigen attached to the well, and then you are going to remove the free antibodies by the washing, and then you are actually going to put the secondary antibody which is actually going to be coupled to the enzyme, and then the free antibody is again washed away and the substrate of the enzyme is added. The amount of colored or the fluorescent or the luminescent reaction produced is actually a direct measurement of the amount of antibody present or amount of primary antibody present in that reaction. A direct ELISA would detect the amount of antigen onto the plate using the enzyme coupled antibodies, and it is rarely been used. So this is the simple, the basic principle that you are actually going to have the antigen coated wells, then you are going to add the primary antibodies, then you are going to wash, and then you are going to add the secondary antibody. The secondary antibody will have the enzyme to their constant region, and then when you add the substrate, it is actually going to give you the product. Then we're also going to have the sandwich ELISA. The purpose of the sandwich ELISA is to measure the level of antigen. So antigen can be detected or measured by the sandwich ELISA, and in this technique, the antibody rather than the antigen is immobilized onto a microtetraplate plate. So basically you have a microtetraplate plate on which you are going to immobilize the antibody, which is being called as the capture antibody. A sample unknown containing the amount of antigen is allowed to react with the immobilized antibody, and remember this immobilized antibody is called as the capture antibody. And after the well is washed, a second enzyme linked antibody specific for a different epitope onto the antigen is added and allowed to react with the bound antibody, right? So after any free second antibody is removed by washing and substrate is added and the color reaction is produced. The common variation on this assay is the biotin linked secondary antibody, and then you can add the enzyme linked avidin in a additional steps. Sandwich ELISA have the proven particularly useful in the measurement of the soluble cytokines such as TNF alpha and other kinds of cultural supernatant, and they can also be used for the cytokine measurement into the serum and the body fluids. So basically what you are supposed to do is you just first put the capture antibody onto the wells, then you are going to add the antigen, right? And what will happen, this capture antibody is going to bind the antigen, and then you are going to use the another antibody which is going to bind the another epitope of the same antigen, and then you are going to add the substrate and it will give you the color, right? And the amount of color is going to be the direct reflectant of the amount of antigen what is present in that particular reaction. Now, sandwich ELISA is being used to measure the cytokines such as TNF alpha and IL-1, IL-8 and all those kind of cytokines which are being very important for different types of immunological conditions. So what we have done, we have actually going to take you to my lab where the students are actually going to show you a demo video how you can be able to perform the sandwich ELISA and how you can be able to measure the amount of cytokines present in the biological fluid. Remember, in this particular demo, we have not used the secondary antibodies. Instead, we have used the alternatives like the streptavidin avidin system, and that's how we are actually going to show you a much advanced system where we are not using the antibodies, the secondary antibody. We are actually using the more advanced system where we are actually putting the streptavidin and avidin system so that it becomes more and more easy. Okay.
In this video, we will demonstrate how to use a pathogenic infection detection kit and what is the underlying principle of that kit. So mostly the detection kits works on immunoassay. So what is an immunoassay? In immunoassay, we will use specific antibodies like monoclonal antibodies against a disease specific or pathogen specific antigen. Then we will develop with the substrate. So this will give some positive color and that will be detected by spectrophotometer reading. The steps include, first step is we have to coat the plate, polyvinyl chloride plate with the capture antibody, following capturing of the actual antigen, disease specific antigen. For suppose in most of the cases, suppose in viral infection, it will detect the coat protein, and in bacterial infection, the external polysaccharide. These kind of antigens it will detect. Once the antibody is coated on the plate, then we will incubate with the sample taken from patient, either it is saliva or serum sample. After incubation, we will wash properly, then again incubate with the primary antibody specific to that particular antigen. It mostly it should be monoclonal antibody, otherwise the detection is non-specific. In next step, after washing the unbound primary antibody, we will incubate with the secondary antibody, that is ceftriaxone conjugated to HRP. So once the secondary antibody incubation is over, we will wash and add substrate solution. Mostly it is TMB or variants of TMB also available for enhanced chromophoric detection. Then we will read the color development using spectrophotometer. These are the main steps. So in step by step, now we will demonstrate how to perform the immunoassay. In order to perform a immunoassay, we need the following materials. First, we need polyvinyl chloride plate, 96 well plate, which should be flat bottom. Then the other materials we need is coating buffer, which is bicarbonate buffer system with pH 9.6. So once it is ready, you adjusted pH, then we will dilute the capture antibody in the coating buffer, then add 100 microliter, dispense 100 microliter each into these wells. So once the dispense is over, then we will incubate this plate at 4 degrees Celsius preferably, but we can incubate at room temperature also for 2 hours. If you are incubating at 4 degrees Celsius, you have to keep it overnight, otherwise 2 hours is enough. We have coating buffer, so I have dispensed it into reservoir. Then I will take the capture antibody, we have to see the dilution. We have to follow manufacturer instructions for dilution. Otherwise, improper dilution may give false positive results also. or no result also. So once it is over, then we have to dispense 100 microliter into each plate after proper mixing. Once the material is dispensed into plate, we have to cover the this plate with the paraffin or covering plate. Then we will incubate this plate for 2 hours at room temperature. Post incubation, we have to remove the unbound solution. So we will remove that. So next we will wash the plate with TBST buffer, which should be pH 7.4. We have to wash at least 3 times properly. Then we are going to block with the blocking buffer, which contains 3% BSA in TBS. In next step, we will dispense blocking buffer into each well, 100 microliter each. So it will cover the non-specific area where there is no capture antibody, so that reaction is specific to particular antigen. The blocking should be done at least for 2 hours at room temperature or overnight at 4 degrees Celsius. So here we have done at room temperature. Once the blocking is over, we have to remove remaining blocking buffer and wash 3 times and incubate with the primary antibody. So I'm going to do that. In this step, we have to dilute the primary antibody with the assay diluent or in blocking buffer. Then mix properly and dispense 100 microliter each into 96 well plate. And incubate at room temperature for 2 hours. Once the primary antibody incubation is over, we have to remove unbound antibody and wash with the TBST for at least 3 times. So it will remove unbound primary antibody, and in following step we will incubate with the HRP conjugated secondary antibody and incubating it for 2 hours. After incubation with secondary antibody, we have to remove secondary antibody and wash thoroughly. So after washing, we will incubate with the substrate solution, which is usually TMB, tetramethyl benzene. So we will dispense the substrate solution into wells. So once the dispensing is over, we have to keep at room temperature for some time until we can see a visible blue color. Once the reaction is over, then we have to stop the reaction with the 2 normal HCl or sulfuric acid. After 15 minutes, if you observe the plate, we can see the blue color intensity in some of the plates is very high and in some of the wells is very less. So that means whatever the wells it is giving intense blue color, that means the concentration of the antigen is very high. So at this moment we have to stop the reaction, otherwise all the wells may turn into blue color. So we cannot identify positive sample versus false positive sample. So there may be some artifacts. So that's why we have to stop the reaction using 2 normal HCl or H2SO4. We will add 2 normal H2SO4 to stop this reaction. So as we can see, that blue color turned into yellow color. So we have to read this in spectrophotometer at 540 nanometer and 450 nanometer to get absolute values of these things. So this is qualitative purpose as well as quantitative purpose. Qualitative purpose in the sense, if you are using samples from patients, you have to do it in triplicate, and you can just identify whether that particular person is disease positive or negative. In another case, for quantitative purpose, you need to have various varying concentration titrations of the particular antigen which that disease causes. So in this case, you have to dilute that antigen in different concentration, and you have to develop the assay in the same way developed here. So you have to plot a standard graph against the concentration versus the observance we have taken. From that value, you will come to know what is the unknown person's that antigen titrations in his serum or saliva. So that's why it is qualitative as well as quantitative method. With this, we can understand this method can also be applied for detection of various chemicals, various drugs in blood, like drugs used for hallucination purposes and recreational drugs, and also some of the drugs used in the pharmaceuticals.
Now, the fourth one is the competitive ELISA. Okay, so in a competitive ELISA, it's very sensitive, right? Extremely sensitive for measuring the amount of antigen. And this antibody is first incubated in solution with a sample containing the antigen. The antigen antibody mixture is then added to a antigen coated microtiter plate. The more antigen present in the initial solution, the less free antibody will be available to bind the antigen coated well. After washing off the unbound antibody, the enzyme conjugate secondary antibody specific for the isotope of AB1 can be added to determine the antibody bind to the well. In the competitive edge system, the higher concentration of the antigen in the original sample, the lower the final signal, right? So this is what the competitive ELISA. What you are going to do is, first you are going to capture the antigen what is present in the solution using the primary antibodies, and then you allow this to immobilize onto the well, right? And then you are going to add the another antibody, right? So this antibody will bind to this antibody, right? And it will actually going to give you the color, right? Now, once you have developed the antibodies, right? Once you have developed the antibodies and you have actually characterized these antibodies with the help of the different types of ELISA techniques, what you can use is you can actually be able to, once suppose you have developed the antibody and then you would like to use these antibodies for different types of applications, then you can be able to use these antibodies, right? So let's see what are different types of applications of the antibody. So antibody are being used in a passive immunity. So remember, passive immunity means where you are actually going to get a significant help from the reagent, right? So passive immunity involves the transfer of preformed antibodies from an immune individual to someone who is not immune, providing an immediate but temporary protection against the disease. Antibodies are key component of the passive immunity. They are introduced into the person's body either naturally or artificially to help to neutralize the pathogen or to the toxic. So you can have the natural passive immunity, which is happens when you are going to get the antibodies from your mom. So during pregnancy, maternal anti-IgG molecule cross the placenta and protect the fetus from the infections like the tetanus, rubella, and measles. Or you can actually get the natural passive immunity through the breast milk, right? So the maternal IgA antibody in the breast milk protects the infant mucosal surfaces against the infection, especially into the respiratory and the gastrointestinal tract. Then you can have the artificial passive immunity, right? So artificial passive immunity where you are actually going to inject the antisera into the individual. There are several scenarios, like you can have the immune deficiency, you can have the toxins or the venom exposures, or you can actually have the rapid responses to the deadly infections such as COVID-19 or the coronavirus. So when you are going to expose to a pathogen, right, it is actually going to cause a serious illness or death before a person's own immune system can respond. So in those cases, what you are going to do is you're going to take the antibodies from a different person, and that person is so the antibody from the different person is going to give you the temporary protection until your body is ready to fight, right? So you can actually have that, you can do that under the immunodeficiency, which means you might be having some kind of defects because of which you cannot be able to produce your own antibodies in a timely manner, right? So one of the example is the acquired immunodeficiency or AIDS, right? So this is what exactly a very serious problem when the person gets the AIDS, because AIDS virus does or the HIV does not allow the body to respond to an infection to produce the different types of antibodies. Or you can actually get the exposure to the venom. So venom actually acts so fast that before immune system could recognize it and produce the antibodies, it actually kills the person, right? So to get the protection against these life-threatening toxins, what you do is you actually directly inject the antibody against the venom protein. Then we can also use the antibody as a blocking agent, right? So antibody can be used as a blocking agent by binding to a specific molecule or receptor involved into a disease process, preventing their activity and thus reducing the harmful effects. So these blocking agents are, antibody, when you use the blocking agent, it probably can take care of the allergies, take care of the any kind of receptor which has been involved into different types of, the pathological outcomes. So you can actually use this for targeting the immune cell movements, or you can also use these blocking agents like antibody interfere with the key inflammatory signals or the immune cell functions, helping to calm down the overactive immune system. And that's how you can be able to manage the autoimmune diseases. Remember, I just said, right, when we were discussing about the antibody development, that you are supposed to remove the self-reactive antibodies, but if you cannot remove the self-reactive antibodies, or some of the self-reactive antibodies are present in your body, then they are actually going to react against your own body antigens. To avoid that, what you can do is just add the blocking antibodies, and they will actually going to block the antigen in such a way that it should be get hidden from the immune system, and that's how it is actually going to give you the temporary relief from the any kind of adverse immune reactions. Then we can also use the antibodies as an anti-cancer agent. So you can actually use the antibody for killing the cancer cells. So antibodies can target the tumor-specific antigens, unique molecule found only onto the cancer cells, and making them a potentially powerful tool to identify and destroy the tumors, right? So you can actually use the active or the passive immunizations, or you can actually be able to use the blocking factors into the serum. So active immunization means the stimulating the body's own immune system by giving a tumor antigen, hoping it protects in own tumor, into tumor response. Or the passive immunization, where you can directly providing the anti-tumor antibodies that are ready to bind to the tumor cells and to help eliminate them. Or you can use the blocking factors. So scientists found that the serum from the animals or patient with the progressive tumor could block the immune responses, especially the killing activity of the cytotoxic T cells, measured by the the cell mediated lymphocytes or CML. So this blocking factor can also just not just the antibody by itself. Often it also the antibody tumor antigens complexes that interfere with the immune attack. So in these cases, what you can do is you can actually use these kind of blocking factors, which can actually block these interaction of the antibody with the antigen onto the tumor cells. Then you can also use the so what happens that antibody may bind to the tumor antigen and mark them, making it harder for the T cells to recognize and killing the tumor cells. So what you can do is you can just prepare the antibody antigen complexes which will block the Fc receptor on immune cells like the NK cell and macrophages, inhibiting the antibody dependent cell mediated cytotoxicity or ADCC, which is going to another mechanism for killing the tumors. So despite these challenges, some clinical treatment today still use the antibodies for the cancer therapy, but they can often not aimed specifically at tumor specific antigen because of the risk of these blocking effects. Then you can also use the antibody for performing the different types of experiments. So you can use the antibodies for the immunohistochemistry so that you can be able to use that for different types, doing the different types of experiments. So immunohistochemistry is a laboratory technique that uses the antibodies to detect and visualize the specific antigen in the thin slices of the tissue, helping the researcher and the clinical study where particularly molecules are located onto their biological context. So you can actually use the antigen antibody interactions or you can use the enzyme coupled antibodies for these particular applications. Then you can use the fluorescently conjugated antibody so that the antibody is whatever the signal comes, it's actually going to give you a glowing, right? You might have seen the immunolocalized antigen onto the thin slices using the immunohistochemistry, or you can also use the direct and indirect detection approaches. You can use the secondary antibodies and so on, right? And then in the immunohistochemistry, the antibodies serve as a precise targeting tool to detect the antigen in the tissue sections. When linked to the enzyme or dyes, they allow the visualization of the exact location of these molecules under a microscope, providing insight into the tissue structures, disease status, and the protein localizations. Then you can also use the antibodies for targeting the antigens, right? So you can use the monoclonal antibodies, which are powerful in cancer therapy because they specifically recognize and bind to the specific antigen onto the surface of the cancer cells. This makes them ideal targeting the antigen, meaning they deliver the therapeutic agent precisely to the disease cells while sparing the normal or the healthy cells. There are two main types of monoclonal antibodies. You can use the unmodified antibodies or you can use the conjugated antibodies. So in a unmodified antibodies, you are going to just simply use the antibodies and they are actually going to work. So for example, like in a B cell lymphoma, the cancerous B cells are expressing a unique antibody on their surface. So scientists can generate a mouse monoclonal antibody that specifically recognizes idiotypes. So when injected, these monoclonal antibody bind only to the lymphoma cells and activate the complement system, which leads to the cell lysis and leaving the normal cell unharmed. Similarly, you can use the conjugated antibodies. So in this case, you can actually be able to couple the different types of drug molecules, or you can use that for killing the cancer cells. So to enhance the killing power, monoclonal antibody can be attached to toxins, radioactive isotopes, or the chemotherapy drug, and this approach turns the antibody to a guided missile. So it carries the toxic agent directly to the cancer cells. One of the example is like radioisotope conjugated monoclonal antibody like the ibuprofen, right, delivers directly radiation specifically to the CD20 positive B cell in a non-Hodgkin lymphoma. Similarly, you can use the immunotoxins where a toxin is linked to the antibody and that kill the tumor cells without damaging the normal cells. And this targeted delivery is crucial because it minimizes the collateral damage to the healthy tissue and focus the therapeutic attack onto the tumor, improving the treatment efficacy and reducing the side effects. Then you can also use the antibody as an enzyme. So you can actually use the antibody for behaving like an enzyme. So the antibody with an enzyme-like activity are referred as the enzyme. Enzymes are a specialized type of antibody that act as an enzyme. They don't just bind their target but catalyze the chemical reaction by stabilizing the transition states, just as the natural enzymes do. So normally the monoclonal antibodies are used for their specific binding abilities and they can precisely recognize and attach to the target molecules. But researchers realized that this precision binding can be harnessed for more than just recognition. So scientists can design the antibodies that bind specifically to the transition state of a chemical reaction, and by doing this the antibody lower the binding energy barrier and speed up the reaction just like the enzyme does. So this enzyme-like antibodies are called enzymes, and they are novel tool because they combine the specificity of antibody with the catalytic activity of the enzyme. So this is all about, we have discussed so far about the antibodies. So we have discussed about what is the structure of the antibody. We have discussed about the diversity of the antibody and how the diversity of the antibody is being produced by the clonal proliferation and selections by the antigen molecules, and then how these antibody can be produced into a animal system, right? So we have shown you a demo video where the students are actually explaining how you can be able to produce the antibodies into the rabbit. Subsequent to that, we have also discussed about the antibody and antigen interactions and how the antibody interaction can be exploited to design the different types of immunoassays. So we have discussed about the precipitation reactions, we discussed about the acutylation reactions, and then we also discuss about the enzyme-linked immunosorbent assays or ELISA assays. So within the ELISA, we discuss about all the variants, right? So we have discussed about the direct ELISA, indirect ELISA, sandwich ELISA, and the competitive ELISA. While we were discussing about the sandwich ELISA, we have also taken you to my lab where the students have shown you how you can be able to do the sandwich ELISA to measure the cytokines from the the culture supernatant. And then subsequent to that, we have also discussed about the different applications of the antibody. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more aspects of the immunology. Thank you.